Good morning, everybody, and a warm welcome back here to Baltic Breakfast. Today is going to be about the smallest creatures in these oceans, microbes. Pretty difficult topic. I'm so glad to see so many of you here. My name is Gun Rudqvist, and I'm a part of Stockholm University Baltic Sea Center, and we are the ones organizing these monthly Baltic Breakfast, touching upon hot topics from a research perspective, but also interesting in a policy landscape. You know, as you all know, we will try to sort of answer all the questions that's popping up during these 45 minutes after two brilliant presentations from researchers, as always. You can either post your questions via email, ostersjocentrum.su.se, or you can go to a Slido code, which is 22102. And if we don't have the time to get all your questions and answer them, we will try to write answers and put them on the web afterwards. So today's topic, microbes. You know, not easy, small and difficult to study, but central to a number of functions in the sea. There's a growing knowledge about these small creatures, and they can also be a pretty powerful tool for environmental monitoring, but we will hear more about this later on. And of course, it touches upon indicators in the Marine Strategy Framework Directive, and that could be things like seafloor integrity or biodiversity. So there is definitely a policy aspect to this. Today, we have two excellent speakers with us, as always. Today, first out is Emma Bell. She's a researcher in environmental microbiology at the Baltic Sea Center, but also affiliated with the environmental genomics group at Sci-Fi Lab here in Stockholm. And after that, we have a colleague of mine, Alexis Fonseca. You are a marine biologist focusing on marine bacteria and microbial communities in sediments. You are currently a postdoctoral researcher at both at DEEP and in Stockholm University and the Baltic Sea Center. And both Emma and Alexis are also part of the larger research collaboration COASTCLIM, which Stockholm University has with Helsinki University. So Emma will start out and sort of more introduce the whole concept and focus on the water column, and then with Alexis speak, we will dive into the sediments. So please, Emma.
Good. And very nice to see so many of you here interested in microbes on a cold January morning. So today I'm going to talk about microbes in the Baltic Sea and how we understand them using DNA sequencing approaches. But first, I want to begin with why should we care about microbes at all? So Earth is a microbial planet. It's estimated that there are 10 to the 30 on Earth. That's 30 with that's 1 with 30 zeros after it, or 1,000 billion. For comparison, there's about 8 billion people on Earth, so microbes outnumber us by more than a trillion to 1. But it's not just the sheer number of microbes that make Earth a microbial planet. It's because microbial metabolism makes the Earth habitable. They produce oxygen, they recycle nutrients, making them available to plants, and they even help regulate the climate. Microbes not only outnumber us, they outweigh us. So all life on Earth is carbon-based. Another way to look at abundance then is in terms of carbon biomass. So this square shows all carbon biomass on Earth, and we can see that microscopic bacteria are second only to plants in terms of carbon abundance. This is then followed by other microscopic organisms, archaea, protists, fungi, and animals make up a very small percentage of that. If we zoom in, humans are here, and you can see that we're only more abundant in terms of carbon than wild animals, wild birds, and nematodes. Microbes are also the most diverse organisms on Earth. So this here is a tree of life. The eukarya, shown in green, this is all plants, all animals, humans, fungi, are just in this small green square. Everything else on the tree of life is bacteria or archaea, so microscopic organisms. And the diversity on the tree of life is reflected in the diversity of function. So every environmental place that we find them, they have a specific metabolic niche. One of the things that I really like about this tree of life is that the author illustrated each of the branches for the life that we have been able to see. And if we zoom into the bacteria, for example, you'll see that most of the branches just have this small DNA symbol, which means that we've never actually seen them or been able to grow them in the lab, but we know about them from DNA sequencing. So what is DNA sequencing? Well, this is a method that we commonly use to study invisible life. We take DNA, extract it from the environment, so in our case this would usually be seawater or sediment, and then sequencing, very simply, is just the order of bases in DNA, A, T, C, and G. And within those 4 letters encodes everything there is to know about an organism. We typically then take one of 2 approaches. We either sequence a single gene that's present within all community members, and this gives us a fingerprint of the microbial community, who is there, or we sequence all of the genes, and then we get whole genomes, and we can start to look at things like function. DNA sequencing is not a new method. So the microbial genomes were sequenced back in the 90s, but as a technology, it's evolved so rapidly over the past decade, along with computing power and the tools that we have to analyze the data, that it's now possible to quite routinely, relatively cheaply, sequence thousands of microbial genomes from a single. So this is one of the tools that we use to study the marine microbiome. This graphic is showing seawater and the abundance of different life in the sea, and we can see that 2/3 of life in the ocean is microbial. So the right 2 are plants and animals, and then everything else is microbial life. And they carry out really important ecosystem functions in seawater. They perform photosynthesis, and they're responsible for producing as much oxygen as plants on land do. They consume methane, so they help regulate how much of this greenhouse gas reaches the atmosphere. They convert carbon dioxide into biomass, which helps the ocean act as an overall carbon sink. And they degrade organic matter, recycle nutrients, and can even help clean up. Microbes cycle carbon and nutrients. This puts them at the base of the entire marine food web. So the carbon moves up the chain that we start with the microscopic organisms. They're then eaten by zooplankton, and the energy flow moves up the food chain. So the marine microbiome is important for the ecosystem as a whole. Which microbial communities are present and what they do depends very much on their environment. So in the Baltic Sea, we have gradients in salinity, we have gradients in light and temperature with depth and season, and also gradients in nutrients. And since the microbial communities are very tightly linked to these conditions, we asked whether microbial communities can also be used to predict their environment. So this was a project that we did as a team in the environmental genomics group, where we took DNA from microbes in seawater. We then combined this with environmental measurements from 18 stations around the Baltic Sea. And we used this information to train a machine learning model. So this works where we have the microbial DNA. We then have abundance of microbes in the sample. We have all of the chemical measurements from the environment, and we give this information to a model which then learns the link between the microbes present and the environment. Why is this useful? Well, we can then give this model a new sample. So we have our DNA, we know the microbes present, we give it to the model, and then the model can tell us the surrounding environment. So in this example here, we have real data on the x-axis, and we have model predicted data on the y-axis. And what these graphs are showing is that the model can accurately predict the environment. So DNA can be used almost like a biosensor, where the status of the environment is recorded in the microbial DNA. And we did this for many different parameters, not just for salinity and nitrate. And we were even able to use bacterial abundance to predict the presence of phytoplankton and zooplankton. And this is because microbial communities are not only shaped by their environment, they're also shaped by their interactions with other microorganisms living in the same space. And these interactions also include viruses. So bacteria in the Baltic Sea are also infected by viruses. When this happens, they inject their DNA into the bacterial cell. They use the bacterial machinery to replicate and then burst out of the cell, ready to infect more microbial cells. This process is estimated to kill 20 to 40% of microbial cells every day, and it alters the carbon flow in the system, because the carbon that comes out of the microbial cell can then be a food source for other microorganisms, or it can form aggregates and sink to the bottom of the ocean, exporting carbon to the sea floor. And this doesn't just happen on the scale of a single cell, like I'm showing here. It happens on the scale of the ecosystem. So this is a picture of a cyanobacterial bloom taken with a satellite. It's from July last year in the seas just southeast of Stockholm. So this is when cyanobacteria grow in such great abundance that we can even see them with a satellite. And you can imagine then if a virus infects this population, causing the bloom to collapse, that this would have an impact on the ecosystem and on carbon export. But before we can understand how this impacts the ecosystem, we even need to know which viruses infect which bacteria in the Baltic Sea. And for this, we then again turn towards sequencing. So for this, we use whole genomes of bacteria and whole genomes of viruses, and we look for a specific region within the genome called the CRISPR array. So you may have heard of CRISPR, Nobel Prize in chemistry, as a gene editing tool, but it actually comes from microbes, where they use it as a defense mechanism against viruses. So how does this defense mechanism work, and how does it help us link viruses to their microbial hosts? Well, when we sequence this region of the genome, it looks something like this. And what we can first see is that there's a repetitive sequence. This is specific to each microbial host. So, for example, I could know which cyanobacteria this sequence belongs to. Then, the short pieces of DNA in between, they belong to viruses that have interacted with this host. It works as an immunity or a defense mechanism because the next time this bacteria sees the same virus with this matching piece of DNA that it has stored in its genome, a protein complex will come and work like scissors to cut up the viral DNA, and then it'll stop the infection. So this is a fantastic form of immunity, but it also leaves a record in the DNA for us to be able to see which viruses have infected which bacteria. And this is an approach that we are using currently to understand the interactions between bacteria and viruses in the Baltic Sea. And there is still so much more information in microbial DNA that I haven't had time to talk about today. But I would like to leave it by saying that DNA sequencing is a powerful monitoring tool. From just 1 litre of water, we can learn so much information about the ecosystem. We can see the microbial communities that are there. We can see the status of the ecosystem and even predict how the ecosystem might change. We can see who they interact with, and we can see how microbes drive ecosystem processes. Thank you very much for your attention.
Thank you, Emma. Thank you so much for clarifying these complex issues for us. So you mentioned that it was sort of new technology. What do you mean by new? I mean, what are we talking about?
I mean, the technology itself is not new, but just the scale at which we're able to do it, and for the cost for how much DNA data you get, and then of course computers being so much more powerful. It's a rapidly evolving field. There's new tools coming out every day. It's
So that means that what you ended up with here, the possibilities for environmental monitoring, do you see that as something coming in the future?
Yes, I think it's a really useful monitoring tool, and that more people are starting to adopt it.
Okay. Is it already being used?
I don't think it's being used as standard, but there's more interest in using it. And like the data that I showed of the predicting, is people had already collected that data, so it's becoming more increasingly used.
Thank you. I'm sure we'll get back to this. Thank you, Emma.
Thank you. Thanks.
So next, Alexis Fonseca, also from the Baltic Sea Center. And we will move now and talk more about bacteria and viruses, but move into the sediments. Alexis,
yes.
please.
Hola, hola. Okay.
Yes.
Well, good morning, everyone. Yeah, so I will address microbes, but from the sediment perspective. So I entitled this Microbial Life in Marine Sediments, Viruses, Bacteria, and Their Impact. I will navigate through 2 main ideas. So I will tell you first how small animals interact with viruses, and a very overlooked interaction, and how this can influence geochemical cycles. And secondly, I will jump into the west coast of Sweden and south of Finland, and tell you about some very special bacteria called cable bacteria in a very anoxic place in the west coast of Sweden that in some way resembling windows to the past and analogs for the life beyond Earth. So the first one is how this fauna interact with viruses in the Baltic Sea and the consequences. So first I will say that we have basically 2 kind of viruses, and the difference is what kind of material they have in their genomes, if they are made up of RNA or DNA. So for RNA, like a COVID, for example, that is we are very familiarized. In aquatic system, they are more prone to infect eukaryotes, like a fish, mammals, birds, or even algae. But DNA viruses are more prone to infect bacteria. That is called bacteriophages or phages. So for bacteria, actually, it's a pandemic every day, and they don't have social distance. So it's more difficult, and this has been happening for billions of years. So they need to develop very good defenses. If we look closer into the DNA viruses, phages, they act as a double-edged sword. So for one side, they can kill bacteria. They infect one bacteria. They assemble the new army. And when they release the army, they disrupt the host. And as a consequence, they modulate the communities and populations and also release tons of nutrients. But in the other way around, they can endow a kind of superpowers to bacteria. Imagine, for example, methanotrophic bacteria in the Baltic Sea, bacteria that can oxidize methane. They eat methane, and they prevent that methane-producing sediments goes into the water column and atmosphere. So if some virus that contain genes, because viruses carry genes as well, infect this bacteria, they can incorporate new genes for methane oxidation, so these bacteria receive a gift, and they are more efficient than to oxidize methane or sulfur. So this impact as well in the cycles. in the biogeochemical cycles. Another force very important in sediments for shape microbial communities, and particularly bacteria, is bioturbation. And what is bioturbation? It's a physical disturbance created by animals in fauna. Here in our project, we divide, we focusing in two layers of in fauna. The first one is microscopic animals, called in fauna. Oh, okay, sorry. Like these nematodes, they are microscopic and the size is under 1 millimeter. Larger than 1 millimeter is microfauna, like a Macoma baltica that is very common in the Baltic Sea. So all of them make up in fauna. And with their daily activity, eating, commuting, burrowing, they create this physical disturbance, move particles, also create holes that permits that oxygen and nutrients penetrate into the sediments, and this create a big impact in bacterial communities because they change the conditions, the pH. And so for some population, this could be very beneficial, but also for some of them could be suffer detrimental effects. A good example is a publication from 2 years ago from Broman and colleagues from the Baltic Sea, and they demonstrate how bioturbation can impact methane in the Baltic Sea. So we know that bioturbation impacts profoundly bacterial communities, and our question was, well, does in fauna bioturbation affect sediment viruses? We took advantage from the previous experiments from Broman. They created basically a gradient of animals. So they put a very high abundance of these animals in fauna and a very low, create these gradients, and also on a portion of natural community to reference. So by the way, these were sediments collected from the south of Finland. And after 10 days, we collect everything, but particularly we collect DNA and RNA from the first centimeter of these incubations. And then we got metagenomic data, metatranscriptomic data to build the RNA viruses and DNA viruses community, and also bacteria, and then analyze abundance, diversity, and activity. What discovered was that actually in fauna significantly affect DNA viruses. So as with more animals we have, the DNA viruses are more abundant, more diverse, and also more active. Opposite to RNA viruses, nothing happened. So then we have a differential result. And it was something that is very new. Nobody before noticed if these very different layers of organisms, animals and viruses can interact. What is the implications? Well, this is like a domino. So we know that bioturbation can impact bacteria, but also we know that they can impact viruses, phages, viruses that infect bacteria. So my message here, my point is we should integrate also these phages into the budgets for geochemical cycles. So now I will jump into a different topic. This is a very special bacteria, cave bacteria, that we found in the west coast of Sweden in a very anoxic sediment, anaerobic environment. And this resembling in some way a window to the past and also analogs for life beyond Earth. So the first idea that I watch away is that bacteria is not just small and unicellular, because actually they can be big and multicellular. In the right, we have these long filaments, like a hair, and this is actually just one bacteria, well, several bacteria, in a scale of centimeters. In the right side, we have an electron microscopic picture, and we can see the morphotype, the shape. We can see some junctions. Between this junction is one cell, and also some ridges. That is very important with this bacteria. So just a reference, one standard unicellular bacteria goes between 1 and 10 micrometers, but here we have on a scale of centimeters. Here I have a video that I want to share with you. So you can see these filaments, very big, and the black dots surrounding these very big filaments are unicellular bacteria. So we can appreciate how big they are. But this is not the main characteristic, actually. Why these organisms, capable bacteria, are unique is because they are the unique organisms that can conduct electricity in a scale of centimeters. And why they do that is because they respire oxygen, primarily, that is in the very top of the sediments, and they eat sulfur. They make sulfide oxidation, and sulfur is just a little deeper into some millimeters or centimeters. So the problem is what they respire is in a different layers of what they use for it. So they can connect one extreme to the oxygen and the other extreme with the food, with the sulfur. They take the electron from the sulfur and conduct this, creating an electric circuit. So they create a different electric field and pH, and also remove sulfur from the sediments that is toxic for other organisms and prevent this goes into the water column. And how they make that is because they have special structures. In these ridges we have nanowires or some fibers that conduct the electricity. But also this is interesting because they have some potential applications. So 5 years ago it was discovered that they make interesting symbiosis with plants, aquatic plants like rice, and they prevent methane goes up. Also, from more engineering perspective, is a lot of people working into this material that conduct electricity because it's very efficient. So some of these people envision that maybe very soon we are going to have electronic devices on base of bacteria, like a cell phone maybe. This is very desirable. So this material will be sustainable, biodegradable. But my interest, and our interest, is more from the ecological perspective. So we studied some stations that we studied before in the west coast of Sweden, Kristineberg Bay and Koljefjord, and 2 stations in the south of Finland. And we use RNA sequencing and we measure chemicals. And what we discovered was something unexpected, because there is a kind of paradigm for these bacteria. They all the time are found on the top of the sediments in which oxygen is present and also sulfur. But in Koljefjord, a very anoxic place, we found this bacteria inhabiting very, very deep, under 20 centimeters. In the beginning, I thought maybe this is wrong and the data is incorrect. But now we made further analysis and this is correct. And probably they confirm some kind of symbiosis with other organisms that produce something that they can respire, something different from oxygen, like a nitrate. Some interesting ideas, just to trigger your imagination, is if we have this community, an anoxic community that get energy from inorganic sources, this is really resembling analogous to primitive life. So oxygen appears 2.5 gigayears ago. The Earth is around 4.5. So before the oxygen appearance, organisms just respire something different from oxygen and get energy from inorganic sources. But also, people that is looking for life beyond Earth, they have very good template here, like in Koljefjord, for example. This is called astrobiology. So a lot of people think that we are going to find this little green man out there. I thought the same, but now I changed my mind. I think probably we are going to find this kind of community, anoxic communities that respire something different from oxygen and get energy from inorganic sources. Just I want to thank, this is a collaborative work, so I want to thank to Coastclimb, to the Baltic Sea Center, and the Nascimentos Lab, and Christoph Humber as well, and a lot of people. Thank you for your attention.
Thank you, Alexis. This is fascinating. We're moving from sort of waters of the Baltic Sea to outer space. Wow. Yeah. So you stressed the fact that this new information regarding biogeochemical cycles, I mean, like the cycling of nitrate, the cycling of methane, etc. So would you actually say that this new information would change the way we look at this, or no?
Yeah, it's the point is, especially with viruses, is we know that obviously bacteria has a profound impact in biogeochemical cycle. Now with the new tools, we can study viruses as well, viruses that infect bacteria. And as Emma mentioned, and I show, viruses can kill, modulate some bacteria. Imagine methanotrophic bacteria. If they just wipe out this population, methane will go out from sediment to water column. So everything is connected. Everything is more complex than we thought. The thing is now with the tools that we have, we can study better these very difficult entities like viruses. So we are getting more information, and for sure they have to say something in these big cycles as well.
Okay, so we should consider them more when sort of
Yes.
dealing with this. Okay, please, Emma, come and join us here. And Alexis, come please. Nej, vi brukade bli så snabbt. Okay, please join us so that you can see them in slide. All right, so now we will open up for questions, of course. I'll get started, of course. So you mentioned, Emma, that the microbes are different in different parts of the Baltic Sea, depending on salinity, etc.
Yes.
So is it also that we know, a bit more about these ones, but not about these guys? Has it been studied sort of equally around the Baltic Sea, or?
That's a good question. I think for sure then there's certain areas like where we have monitoring stations, research labs that will have many more samples from that place. The Baltic Sea, I think we've got a relatively good coverage, but of course if you take a sample in one place, it's so big we could learn a lot more.
Yeah, even though, as you said, we could learn more by using these new technologies. Yeah,
Yes.
but we still need to take the samples. We can't skip taking the samples.
No, taking the samples is crucial, of course.
So why are these crucial?
So that we can understand the changes in the environment, the different microbes that are present at the different parts of the environment, how they impact the ecosystem. If we have gradients in nutrients, what metabolism are doing that changes the system. This impacts things like eutrophication or nutrient loads. And we need to have the microbial DNA to be able to start to understand these processes.
Yeah, and then add the viruses on top of this, right?
Yeah, as you mentioned before, is they can also it's interesting that viruses can endow these superpowers. So also some particular populations of bacteria, for example, can receive some boost, and so they impact also in some cycles, like a sulfur in sediments, carbon, or methane that is important here in the Baltic Sea.
Okay, thank you. Questions, please. Okay, sorry. No, let's start here in the room. Next, yeah.
Thank you very much. I'm not a biologist, so my question may be a little too sort of beginner, so to say. But I have a question for Alexis. It was very interesting to know about that the virus can kill the bacteria, and that could really change the ecosystem. And are there kind of, in a bigger way, explosion of any viruses that could change our ecosystem in a big way, or is this some kind of a natural process that has been going on years and years?
I think it's a natural process that they have been happening for just billions of years. So from when bacteria appeared, probably viruses appear as well. So it has been an endless war in which they try to surpass. So the unique thing is now we have more resources, we have better tools, we have more data, so we can study better these kind of very elusive organisms. Well, it's a debate if viruses are really organisms or not,
Okay,
but this is another discussion. So yeah, this is something natural.
So we're learning about something that's been existing for quite some time.
Yes.
Yeah, okay. Lisa, from the web.
Yeah, from the website we have a question. How does anoxia in large sea bottoms, like the dead bottoms, affect the microbes and their role in the ecosystem?
Okay. Do you want to go first, Emma? Yeah.
Sure. I mean, it's usually the microbial processes that are actually making the sea bottoms anoxic in the first place, because different loads of organic matter means that they use the oxygen, and this is what creates the anoxia. And it's different groups of microbes that use oxygen or not. So I would expect to see different microbial communities there.
Do you want to add something? Or?
Yeah, I think anoxia is an increasing problem. And from the sediment perspective, oxygen is totally crucial. So you can differentiate communities, and when I talk about these cable bacteria, some good aspect, if, for example, they can oxygenate a little more the environment and also remove sulfur that is toxic. And when we have anoxia, sulfur is very abundant. And actually, this can kill some.
So the cable bacteria will be our helpers.
Maybe it can help
okay,
in
great.
some way.
Please, over there.
I have a curious question to both of you. Now we have for about 2 decades basically the ability to sequence a lot of DNA from the environment. Do you actually expect to see traces of some kind of evolution during those 2 decades? Because you said microbes, they are sort of a tracer for the environment, are linked to it. Do we expect to see some traces that they actually adapt during that period?
Definitely, yeah. They adapt on much shorter timescales than we see evolution. So this is one of the reasons that not only taking microbial samples is important, but having the long-term time series so that we can see the changes in the community, we can see the changes in the environment. And the virus project that I talked about, I didn't go into the results, but this is what we were really looking at to see how the bacteria and viruses evolve together over a 10-year time frame from this monitoring station near Kalmar. So yes, we can see evolution on a smaller scale.
Okay, interesting. Any more questions? All right, so yeah, please here, Helen.
Thank you very much. I was just curious because you were going to the sci-fi here. Would you think that we could use microbes? Because now when we try to make the Baltic Sea more healthier, we are often going through the perspective of changing us, of course, and we should. But would you think, if you go a little bit beyond where you are right now, that we could start to use microbes as a way of treatment as well? I mean, for dead sea bottoms and such.
Okay. We're getting back to that. Are they being our helpers? Okay.
Yeah, I think so. I mean, bioremediation itself is already a field. For example, with oil spills, you can use microbes that start to degrade the oil. So absolutely, I think we can look towards microbial metabolism for solutions to environmental problems.
You agree, or?
Yeah, I think I just recently I read about something of these big bacteria. Just to be clear, this big bacteria like cable bacteria, just one of these bacteria into a big group. There are basically they eat sulfur, and I know that some experiences using this kind of bacteria to try to remove this sulfur that is very detrimental for other communities. So, and also I know that for particularly this cable bacteria, there is a big project in Europe in this symbiosis with plants to try to remove, for example, methane. That is one of the increasing problems with the climate change.
More questions from the web or from the net? No, not so far. So, oh, sorry, I didn't see.
I was just thinking about there are some new poison things from läkemedel,
Pharmaceuticals?
Yeah, yes. And they get into the environment, into the sea. Do that kind of thing affect the microbes?
Oh, I was just about to ask the same thing. Should we take care of the microbes since they are so key from a functional perspective?
Absolutely. I mean, there's often the analogy that people are quite familiar now with thinking about our gut microbiome and how it's so important for our health. So we can think of the marine microbiome in the same way that looking after the microbes in the Baltic Sea is very important for the ecosystem health.
So what could that be, looking after? Is it possible already now to sort of specify that? What would that be?
I don't think I can give you an answer right now of exactly how we should look after them, but it's the same way, you know, if you eat rubbish, if we put rubbish into the sea, it changes the microbiome and it has an effect on the ecosystem, and then the ecosystem is less healthy because of what we put into it.
So we're getting back to that, having a healthy sea in general, sort of. Would you agree, Alexis?
Yeah. No, just recently with some people from deep and the Baltic Sea, we published an article, for example, related with copper contamination in the Baltic Sea in 2 areas, in Oskarshamn Bay in comparison with ASCO laboratory data. And we can see that how with this Ari and Oscar sham that have been submitted to this pollution, the bacterial community are different, they evolve. And also you can use this like in some way like a sensor. So we can, in some way we can see also the bacterial community, if they change because it's pollution, they make a kind of community services because they bring some nutrient and stuff also to environment. So, yeah.
Yeah, okay, so they should get paid for their ecosystem services, right? Yeah.
Absolutely.
So if we talk about biodiversity in general, I mean, we talk about terrestrial biodiversity and the importance of maintaining it, and also in the marine areas, etc. Is it important to think about microbial biodiversity from a functional perspective?
Definitely, yeah. When we change the ecosystem, we change the microbes that are present and then change their function, and that might be a key function or ecosystem service that we need to keep the environment healthy. So we can also experience loss of microbes when the environment changes, and they may have been key to the ecosystem.
Yeah, I agree.
Yeah,
Yes.
you agree? Okay, great. Any more questions or thoughts? Now you have the possibility. Yeah, over there, Barbel. This is... Oh. Ellen gets to run with the microphone so we can hear you.
You guys had this funny little bit bioengineering or can we use bacteria ideas. Could you just think if we were able to prevent bacteria from degrading carbon when we are running the risk of getting anoxic bottoms, what would that mean for the rest of the biogeochemical cycles, for the nutrient cycles, for oxygen budgets, carbon budgets?
So do you mean that if we stop one function, would it affect the others?
Yeah, exactly. What would that mean?
Okay.
I would say that absolutely everything is linked. So before initiating some form of bioremediation, you have to think about how it will impact all of those things that you mentioned, because of course if you change one thing it can have a knock-on effect. So yeah, I think that would need to be assessed before you implemented anything.
So in sort of the daily work or sort of monitoring, etc., we don't presently have any thresholds for microbes, or we don't really look at microbes, or no?
No, I don't think so. So for biology, routinely there's microscopy or doing things like chlorophyll measurements. So microscopic life is considered, but usually DNA sequencing isn't included as standard.
Okay, so should we, or?
Yes.
You should.
You mean to use as a monitoring process? I think in sediment usually is the standard is use fauna, for example.
Okay.
I think there is a technical problem in, about microbes to use as a sensors, because detecting a species, for example, it depends on taxonomic ranking. So species make something different to genera, the up level, and it's very difficult to detect very fast species. So maybe we can detect very fast families, but below family you have genera. Maybe they don't have the same metabolism. So how I can predict?
So it sounds
I
like
think
the
it's a technical
taxonomy is a bit still.
So I think there are some technical problems.
Okay, but
this
that's solvable, I suppose, or?
I guess as technology is just evolving, we are going to be able to solve these problems, and then it will be easier just measure in 2 minutes some bacteria and sensor the environment.
All right, so as always, we finish these breakfasts with the final question about if you can decide, make a decision that has an impact on what you work with, what would be your decision? What would you like to see happen tomorrow?
I would like to see microbes included in monitoring programs.
Okay, that's good. You need funding for that.
Yes.
Wait, I didn't think about that actually. But now, some days ago, I read something about synthetic life, how people is using some large language models to create, for example, viruses. And I would like to go in some direction, obviously to do good things, again, because it's a risk as well, but something like that.
Thank you ever so much, Emma, Alexis.
Thank you.
time is running out, but we'll be back already on February 18th, and we'll talk about seals. And that's going to be in Swedish. Thank you very much for today, and welcome back.